concrete slab. So Drosophila and melanogaster are cool for a lot of reasons, but one of the most classically studied phenotypes for Drosophila historically has been their evolved high tolerance to ethanol environments, which makes a lot of sense when you think about where Drosophila live and reproduce. They live mainly in disgusting places like this compost pile. Their main food source is rotting fruit. So both our adults and our larvae have to deal with that. Drosophila has also, melanogaster, has evolved much higher ethanol tolerance than its sister species. So here circled in blue are some lines Drosophila melanogaster collected in Africa compared to its closest relatives. The Drosophila story gets a little bit more interesting now because Drosophila originally were found in sub-Saharan Africa. It's where they first evolved and differentiated. However, they had an out-of-African expansion, very much like humans, and colonized pretty much every corner of the globe. And interestingly also, these populations in Europe and Eurasia had a secondary migration pattern to North America and Australia. And in regards to ethanol tolerance, what we see is that these populations that have moved out of Africa have increased their ethanol tolerance even further, so further away from their closest relatives and even further away from their own ancestors. So that we see that these derived populations have higher ethanol tolerance than those in the tropics. And what this sets up is a really good natural system for thinking about how plasticity is playing a role in ethanol tolerance. Because not only have these flies moved to new places in the world, as both of the Matthews have set up earlier in this session, they're also encountering new thermal environments and new food substrates. So that's where my research is going to start, by asking two major questions today. One, what is the impact of ethanol on vulnerable components of fitness? And then for the second part of the talk, we'll seriously dive into more of the plasticity, which is what you're here for in a plasticity section, and talk about the plasticity of tolerance across these different environments. And I just want to note that the work I'm doing has been supported by a lot of different research looking at temperature and physiology, development time, and ethanol tolerance, showing that these characteristics are all really linked together. Back to going forward in time. So, to be able to do this work, first we have to quantify variation in thermal plasticity for both larvae and adults. To do this work, I graciously got lines from Andy Clark's lab. These lines have been brought into the lab and have been inbred, but originally they were from Norway and from Ithaca. So these lines represent those temperate-derived populations which you would expect a priori have very high ethanol tolerance. So, to measure larval responses, I put ethanol into their food and transferred them onto this food when they were third net stars. And then we looked at an array of different phenotypes, survival, energetics, and development. Adults are a little easier to measure. You take newly enclosed adults, you put them in a vial with ethanol, and you see who survives. So, what happens when larvae are exposed to ethanol? Short answer is they really like it at a certain point, but there's a much more long and interesting of the classic measurements of looking at any toxin is to look at lethal dose 50, which is what dose of whatever toxin you're looking at is going to kill 50% of the population. So whenever I talk about survivorship through the rest of the talk, those are all going to be LD50 numbers. And what that means is, for in our case, IO7, if we're looking at incubation time, the dark colors are for 25 degrees Celsius, the light colors are for 16, that they were tolerant of ethanol until you got to about 12%, and then 50% of the population started to die. And one major pattern that we're seeing in this line is that the larvae are more tolerant of ethanol when the temperature is warmer. So this is only putrefaction. If I wanted to look at who actually enclosed us and who survived putrefaction was able to complete metamorphosis, we find that there's a significant drop in that ability, which meant that larvae who went to pupate didn't necessarily have enough energy to complete metamorphosis and enclose as adults. And in fact, a lot of these pupae, you could see them stopping to develop and starting to decompose in their pupal cases. Yeah, it's very sad. And interestingly, in this line, we didn't see a difference between males and females. So now to show you the rest of the data, we see that actually IO7 is pretty typical compared to the other ones. But since I'm in a plasticity section, we're all trying to see plasticity. I'm sure a bunch of people have already been like, oh, but look at I-23. So I-23 is showing some interesting plasticity where they're more tolerant at the cooler temperatures. And this is beginning to give us a hint that even though, yes, larvae don't like ethanol and they're having trouble actually completing metamorphosis beyond just pupation, that there is a piece of genetic variation and possibly even some G-binding interactions happening here. But while I'm talking about the energetics of completing metamorphosis, let's think a little bit more about energetics and what's going on at the physiological level with these larvae. So a good, easy way to measure energetics in Drosophila is to do pupation height. So when a larvae decides to pupate, the first thing they'll do is they'll crawl away from the food. And the idea here is that the further they can crawl, the more energy they had to do this, the more energy they essentially have to spend. This is actually relatively easy, if not horribly boring to measure, because all you do is you divide your vial into 5 quadrants, and then you simply count the number of pupae in each quadrant to come up with an average pupation height. So, what this looks like in the case of our data, when we're looking across this ethanol gradient, is that as you increase ethanol, pupation height begins to decrease. And there's an interesting breakpoint here. You can see that from most lines, their energetics become compromised around 9% ethanol, which, unless you have a wonderful memory, is pretty much the common LD50 for a lot of those lines, hover in that 7 to 12% range. So what that means is, as they're starting to be stressed by ethanol, they have less energy to spend on their pupation height. That's a pretty simple height. Another good way to look at what is going on with these larvae and their fitness traits in energetics is to also look at development. And what we see here is that the ethanol in their diet is significantly extending their development periods. The more ethanol you have in the diet, the longer it takes a larvae to successfully complete development. And for females, what I found was that there's an effective environment. Obviously, ethanol and temperature both having major effects on development time. If you look at the y-axis, you can see that these 16-degree flies take anywhere from 550 hours to close to 700 hours to complete development time. If you work with Drosophila, that feels like forever. While the ones raised at 25 are developing somewhere between 200 and 280 hours. There's also a genetic component here. So with the other things that I've shown you, our two populations have been indistinguishable from each other. But female development time, we're seeing a difference between the populations, with the populations from Norway having a quicker development time than those from Africa. Now, things get really interesting though when we look at males. have males, those population differences go away. But what we start to pick up are these big G by E interactions. I know the plot just got a lot scarier because I added all the lines instead of just the populations. And you can also see that it is essentially a mess of crossing reaction norms. So these males, between these two temperature gradients, and even in some lines, they'll change their LB50 between pieces in the gradient. So they'll be fine up to a certain point, and then the slope of their line will change drastically. So that was a lot of information I just threw at you. And if we put ourselves back in the space of what is actually affecting a larvae, what this comes down to is that ethanol does impact their fitness, which isn't surprising, because it affects survival. But I think more importantly, a little bit more interestingly too, it also affects their energetics, their ability to move, and their development time. And why I think that's particularly interesting is because even if you are surviving on these ethanol treatments, if you have compromised energetics, you have more downstream fitness consequences. I can tell you that these larvae were not happy. Like, I would put larvae on 18% ethanol food and they would crawl away from the food, trying to figure out if there was food somewhere else in the vial that they could eat. A major cause of death, actually, around 18 and 22%, is larvae dying on the sides. So, let's think a little bit more about plasticity that I've already alluded to happening. Okay. So, this is just looking at the effects of temperature in each life stage. And again, we're looking at LB50. And what we see here is when we're looking at the larvae, we pick up both effects of genotype and these interesting G by E interactions. However, when we look at adults, their plasticity strategy has changed significantly. They have stronger responses, and they have more consistent responses, but now every line is showing the same response, where they have more tolerance to ethanol at colder temperatures. What I find really cool about this, and something I'm going to be looking into more in my next project, is that larval responses don't predict adult responses. So remember, these are the same genotypes. These organisms have the same genes. And some of the best responders as larvae can become much flatter as adults and will change the direction of their reactions to ethanol across these two temperatures. So just to summarize, larval development is very sensitive. The genotypes vary in their larval and adult thermal abilities to deal with ethanol, which I just showed you in the last slide. Genotype and environment interactions can be sex and life stage dependent. And larval thermal plasticity is not predictive of adult plasticity. And to get a better sense of why that may be, the next project that I'm going to be undertaking in the Monsters Lab is to do an RNA-seq project to look at underlying genome-wide expression plasticity across these ethanol